Welcome, everyone. You're almost graduating from the boot camp, just 4 more hours to go. We'll have the same format as in all the other days. We'll go for an hour and then have a break. Welcome also to those of you on Zoom. I already sent the slide link there as well. I'll repost it again in a few minutes. For those of you in the room, since we're in person again, just raise your hand and Chandler will call on you. If you can, just speak up. The microphones in the ceiling will pick you up, and so that the people on the call can hear you. And if it's quiet, you could briefly repeat the question. That would be great. So first day, we talked about graphical models. The second day, we talked about causal discovery. The third day about potential outcomes. And so today, on our last day, we're going to hear about experimental design. After that, we've of course covered all of causality that there could possibly be, and there's nothing else to be said. So you will have fully learned everything there is. Of course not. There's much more, and we've had to leave out a lot of stuff that we can't cover in the boot camp, but that's what we've got the rest of the semester for. In any case, thank you very much to Chandler Squires, who's a grad student at MIT in computer science, for taking this last day through the experimental design. Over to you, Chandler. Thanks.
Great. Yeah, and thank you to all the organizers, Frederick especially, for having me talk today about this. It's a really fun topic, so I hope I convey some of my enthusiasm for it. And just kind of as a brief note before we begin, I'm going to mostly be talking about experimental design for causal discovery or causal structure learning. So kind of the same goal of learning a causal graph as we had on day 2. And in particular, picking interventions in order to learn this causal graph so that we can kind of fully identify the system. And this is just a brief outline of kind of how I want to structure the day. May or may not stick exactly to this division of things, but I'm thinking in the first hour, we'll go through some of the settings, the aspects of the different settings that you might be doing experimental design in. And really kind of nail down and clarify those so that we understand what settings each of the different algorithms are going to be operating in. And then we'll do some preliminaries that kind of builds on some of the stuff that we built on the first and second day in terms of what sort of information can be learned from interventions. In the second block, I'll talk about non-adaptive experimental designs, and I'll kind of more formally clarify that a little bit later on, but that's essentially things where you decide the whole experimental design, all the interventions that you want to do at one point in time. And that's to be contrasted with adaptive experimental design strategies, which I'll discuss in the third block, where you get to observe the outcome of each of your interventions before picking a new intervention. And then I'll finish up with some areas where there's more open challenges. So things like targeted experimental design, where the goal is not specifically structure discovery, learning kind of the whole causal graph, maybe learning part of it or learning a certain aspect of it that you are interested in, as well as a few other open challenges. So getting started off with part 1, the examples and kind of aspects of the problem setting are going to be largely motivated by settings in genomics. So you can imagine inside of a cell, there are a bunch of genes that all regulate each other. And for the sake of assumption, we'll assume that these are actually following a DAG model, which is the stuff that we've been working with pretty much throughout this week. And the types of interventions that we can do on this in genomics, we have these great tools for knocking genes out. So you could essentially think of this as an intervention where their value is set to 0. And we'll go through some of the subtleties of the actual experiments and how knockouts are actually kind of affect the data generating process. But from a very high level, just think of it as setting a gene equal to 0. And here I have, we have these 4 genes. You could have 2 to the 4 different experiments if your only ability is to intervene and knock something out. So you could do any of the kind of single gene experiments, or any of the 2 gene experiments, or 3 gene experiments, or 4 gene experiments. So this is kind of the most general setup. And the question we'll be asking ourselves is, how do we pick experiments to learn the underlying causal graph? And we could probably just intervene on every single thing individually. That's not going to kind of be the most efficient thing. So we're going to be looking at kind of different notions of efficiency or optimality of the experiments that we want to pick. So I'm going to go through some of the kind of limitations that you may or may not encounter in different experimental designs now. So as I discussed earlier, the kind of,
Sorry, basic, super basic. What does it mean to knock out a gene, and what is the meaning of an arrow from one gene to another gene? How does it cause?
Yeah, so I'm going to take the kind of view of not explaining the biology here, but explaining just a kind of structural causal process of, we imagine a computer program where a value is sampled for the first orange gene, and then you go down kind of the causal DAG, and each of the things below it, you sample the value from a distribution that's conditional on its parents. And knocking out is just going to be replacing a line in that program with setting the value to 0.
So are we set it to 0, no knockdowns? It's a binary kind of intervention.
Yeah, I'm going to go into kind of distinctions between different types of interventions in a few slides. But I think this is the easiest thing to keep in mind, just this binary thing. So the kind of highest level distinction is between a non-adaptive experimental design, where I pick, okay, maybe I'm going to do this knockout on the orange gene, and then I'm going to do a knockout on the blue and green gene all at the same time. Other ways that you could phrase this, and that you'll see in the literature, I guess you won't see single round in literature, but you could think of this as being a single round of picking interventions. You'll see passive, because you only get to do something once, and then kind of the rest of the data that you gather, you just kind of gather in a passive way after making that one decision. You could call it parallel, because it's all happening. These don't actually have to happen at the same time, but they could happen at the same time. You could be assigning 3 grad students or 2 grad students in this case to be running these experiments during the same week, or batch, kind of similar. And then the final terminology to take note of is calling this just a fixed experimental design, where you fix it ahead of time. This contrasts with adaptive experimental design, where you say knock out the orange gene, see the outcome on your data, and then after that you decide what intervention to do next. So maybe in this case you make the same decision you initially made, but you're making it with more information. So you could call this, to contrast with single round, you could call this multi-round intervention design. To contrast with passive, you could call it active. And then you'll also see this called sequential. So this is kind of more of the sequential decision-making problem that people might be familiar with from Bandits and other literature. So this is the highest level distinction. I expect to, kind of see this in an abstract, or like kind of look for it. This is the first thing that I'll try to identify. The next thing would be whether the set of experiments that I'm considering is unbounded, so I can intervene on as many things as I want to at the same time, or if it's bounded. This could be either because of some technological kind of constraint, where you have to only intervene on a single gene at a time, say in knockout experiments, or even a kind of constraint like, oh, if you intervene on all the cells and knock them out, or all of the genes and knock them out, the cell's gonna die and you're not gonna be able to gather data from it. So there are kind of practical limitations in terms of the number of experiments, the number of intervention targets that you might be inside of each intervention. The next thing I'll distinguish between is whether we're working with a fixed budget. So you can kind of think of this as an optimization problem, and we'll fill in kind of what we might mean by these different terms, but where you're trying to maximize the amount of info, that your information about the system that you're gaining from your interventions under a fixed budget, where you keep the cost of the interventions lower than the budget. Usually people will consider each intervention costing the same amount. So you're just trying to keep the size, your number of interventions below a fixed amount. But you can also assign costs to specific interventions and try to keep the total below some amount. And this is to contrast with, if I just had a big pile of money and I say, I want to completely identify the system, I have some sensitivity, I do care about how much it costs. So I'm going to minimize the cost, but there's no kind of budget. I'll keep going and I'll keep experimenting until I completely identify the underlying causal system. So this would be like a minimum cost identification setting. This is what a lot of the work is going to be focused on. And then the final experimental limitation or kind of detail that I'll incorporate into this picture is whether or not that there's observational data available before. So if there's no observational data, if you just say, oh, I've never seen this cell type before, and you have to go out and figure out what the connections between all of the genes in the cell type are. You start with these dashed lines are supposed to mean, you kind of start with no information. It could be anything. It could be that they're not there. They could be oriented in either direction. Whereas if you start with observational data, say infinite amount of observational data, this will pinpoint you down to at least the Markov equivalence class. So this is something that we talked about on day 1 and day 2, that from observational data, you can at least get, if you recall, it was the adjacencies between the different genes in this case, or the variables more generally, as well as V structures. So the true causal graph was the one that we had on the slide before. There were no unshielded colliders, no immoralities. So you would just get these adjacencies, and you'd be starting with that type of information. So is everything about kind of the setup and these binary kind of distinctions clear so far? So I'll kind of keep this table kind of going throughout and highlight which setting each of these different algorithms are in. We're also going to, I'm just going to add these additional assumptions, not as kind of a binary thing. It's pretty much all of the algorithms that we're going to talk about in the first 3 parts will be in a specific kind of regime of this. So a lot of algorithm design starts by considering the noiseless setting. That's what we're going to be focusing on today. So you can imagine, like I said, with the observational data, essentially you have enough samples that you're pretty confident, it's as well as like having infinite samples, that you can tell the Markov equivalence class. And then from each of your interventions, your intervention is kind of strong enough, you obtain enough samples that you kind of completely identify everything that can be identified with that intervention. There's no uncertainty left outside of kind of pure problems of unidentifiability between different models. And we'll talk in the kind of 4th part about how to relax that and what people do. Kind of the way that you can think about it now, if it bothers you, is that similar to like kind of the definition of algorithms that we had in the causal structure discovery lecture, where we kind of framed it in terms of a conditional independence oracle. You can think of what we have as what I'm calling a conditional invariance oracle, that allows you to tell whether a conditional distribution, like one node conditioned on some other set, is the same between 2 of the kind of interventional data sets that you have. And in practice, similar to like how in practice with the causal structure discovery, you would be doing hypothesis testing. You can imagine doing hypothesis testing on whether or not these 2 conditional distributions are equal. And like kind of a naive thing that you might initially start by doing is just setting a threshold epsilon, where you're going to keep running and obtain enough interventional samples from each of your interventions so that you can distinguish any 2 distributions that are further than epsilon under this measure, and under some measure of total variation distance. So yeah, throughout, like maybe try to save the questions for like stuff about the noisy setting until later on. And then we're also going to be considering causally sufficient settings. So settings without latent confounding. This is kind of the focus of the first half of the causal structure discovery lecture. So we're going to see what we can do with this. Now, somebody asked about knockdown interventions. This is a type of intervention in genomics where you decrease the expression of a cell, but of a certain gene in a cell, but it might still depend on its parents in some way. You've just kind of decreased the total dependence on its parents. This is a real practical problem in different experimental design settings, that not every intervention kind of looks like this idealized intervention where you just set a variable to some value. And because of the history of this do calculus and the notation of do, we're going to call these do interventions. There's other names for them out there. A little bit more general type of intervention is one that still cuts a variable off from its parent. So at the top, we kind of see what's happening to the graph. But instead of setting it deterministically to some predetermined value that you set, you might be setting it to some distribution. And these are called perfect interventions. So you can imagine in the knockdown experiment, maybe you actually do remove the dependence on the parents or something, but there's still a little bit of noise, like this gene might end up just randomly getting transcribed despite none of its kind of parents like telling it to be transcribed. And then kind of the most general type of intervention is going to be called a soft intervention or a mechanism change. If you think of this line as kind of the causal mechanism which generates X2. So this allows you to retain dependence on your parents, but it just changes it. So, if you're in a linear model, this could be something like cutting all of the coefficients in half, or just adding a shift and taking something down by 1 or 2 or however many kind of units. And for our purposes in this talk, these types of interventions are essentially going to be equivalent. So I'm going to define here the types of identifiability that you can get from different interventions. I'm going to do it through this lens of an interventional augmented graph. So if we have a causal system, X1 to X2 is like kind of the top observational system. And then on the bottom, we've changed the mechanism that's generating X2. We can represent this, if we still think in terms of structural causal models, by a indicator variable, this zeta1, that's telling X2 essentially which of these distributions to take its value from. So it's a 0, 1 variable. If it's 1, then this should actually, if it's 1, then it's going to take from the interventional distribution. If it's 0, it's going to take from the observational distribution. And if we have multiple intervention variable or different interventions, we'll just add 1 for each of the intervention variables, or for each of the interventions. So zeta 1 will be corresponding to the intervention I1, and we'll just add between each of the intervention variables that we introduce an edge to the targets of that intervention. And we're going to call this the interventional DAG or IDAG. So throughout, this kind of can get confusing at first glance when we have a set of interventions, and each of these, if we're just defining it by its intervention targets, kind of forgetting the information about whether this was a do intervention or perfect intervention or soft intervention, then this is going to be a collection of sets. So this calligraphic I is always like a collection of sets in my notation. And it defines this interventional DAG according to this construction. Right, so this kind of construction is maybe a bit more recent way of viewing like what interventions are doing to a system and obtaining facts about Markov equivalence in our interventions and stuff like that. So talking about Markov equivalence, the original definition that we had for an observational distribution was that a single distribution was Markov to a graph if There's many kind of equivalent characterizations, but one thing that we said was that it factorized according to that graph. So you can write out the distribution over the variables as a product of each of the variables given its parents, the conditional distribution of each variable given its parents. So we're going to define a similar notion for now an indexed set of distributions where we get one distribution for each intervention. So the first requirement is that each distribution individually is still Markov with respect to the graph. This is kind of a sensible one. We're not breaking the Markov property when we do an intervention. We might be breaking other things, like we discussed faithfulness. If we're doing a do intervention, it doesn't have to be faithful to the original graph, but it's still going to factorize according to the original graph. This is basically due to the constraint, even in soft interventions, that the variable is still a function of its parents and not other variables. And then furthermore, we're going to impose this constraint that's basically just saying what we mean by the system staying the same between different interventions. So if we have some variable xi that isn't in either of the interventions, the kth intervention or the k prime intervention, then the mechanism that generates it, so the conditional distribution of that variable given its parents, has to remain the same. And similar, we'll build up from this definition of a set of distributions being I Markov with respect to a graph to the notion of an I Markov equivalence class. So two DAGs are going to be I Markov equivalent if every index set of distributions, which is I Markov to one of them, is also I Markov to another. So if you think of this as a set of distributions, these two sets are equal. And then I'll introduce some notation I don't believe we've used before. So we'll be needing to kind of keep track of Markov equivalence information throughout a lot of our algorithms. So this is kind of shorthand notation tells us what the I Markov equivalence class of a graph is. So we'll have these brackets and then subscript with what the interventions are. And then in the interventional case, I'm just going to not have a subscript and just use the brackets, or in the observational case. Any questions so far? So we'll start off with, we had this graphical characterization of Markov equivalence that was useful for us for many different things in the observational setting. We'll also have a graphical characterization in the interventional setting. So we can basically extend the notion of having the same skeletons and same V structures just to the interventional graphs. So the graphs of these augmented interventional variables. And this is saying that the two DAGs are I Markov equivalent to each other if and only if these graphs still have the same skeletons and V structures. Maybe a little bit more informative way to see this for our purposes of like asking, what information do we learn from an intervention is to think of kind of what new V structures each intervention will introduce. So I'll write an example over here. If we have a graph like this, and say we intervene on x2, so I'll add this 0 to 1 variable. Right, so we introduce a V structure between these two distributions here. If you think about this in terms of conditional independence, this is saying x1 is independent of this zeta 1, is not independent of the zeta 1 given x2. Right, so this is, they are deconnected. So generally, this is going to be saying that if we kind of interpret this as this 0, 1 variable that tells us what setting that we're in, it's saying that under this intervention, the conditional distribution of x1 given x2 is not going to be equal to the kind of observational distribution of x1 given x2. So if you think about, this being, say, a perfect intervention, this is pretty clear that if previously x1 depended on x2, now we cut off that dependence, now it no longer it's independent of x2 in this distribution, but not this distribution. And similarly, so we'll get this orientation. We'll also get the orientation between x2 and x3. And that's because of, if you go back to the day 1 and day 2, we talked about meek rules. So these are kind of these logical constraints that preserve the acyclicity of the DAG and like aim to not introduce any new V structures. So there's a meek rule that tells us, if this edge was unoriented, and then we look at x1 goes to x2, and then, well, we can't orient it this way because that would create a V structure between x2 and x3. So we get this oriented that way. And this is like a general phenomenon, even if we have multiple interventions. So if we were to have this variable also intervene here, now we wouldn't be able to tell these two apart, say that we just knock both of these out, set them to 0. There's kind of no way to tell the causality between x1 and x2, but you can still tell x2 to x3 because otherwise you would be creating this V structure. So this is kind of the argument that allows us to conclude that the V structures that are introduced by our interventions will exactly give us the edges between variables that are in the intervention set and variables that are not in the intervention set. So that's just written out here in terms of if we have an edge xi to xj in the first graph, and only one of those variables is in the intervention set, then that edge also has to be directed the same way in the second graph for these two things to be Markov equivalent.
Can you please elaborate more on why f prime and f observation? Yep. Why they are not equal?
Yeah, so maybe this is an issue of maybe what we talked about on the second day of faithfulness. There are ways that you could set this up so that they be equal, but in general, you can choose a distribution such that they are not equal. So say that x1 was, Bernoulli 0.5 and x2 was equal to like x1 plus, or like, XORed with some other, let's say Bernoulli like 0.1 variable. So like 10% of the time, we flip the sign of x1 when we generate x2. So in general, x2 tells you some information about x1, right? So the distribution of x1 given x2 could be given to you by Bayes' rule. And you'd have higher probability of x1 equaling 1 whenever x2 equals 1. But then if I come in and I intervene on x2 and I say, okay, x2 is always equal to 1, now it doesn't tell you anything about x1.
I just want to understand how you draw those zeta variables.
Yep.
In this case, do you draw zeta 1 go to x1, go to x2, or as in this case, you draw just zeta 1 that goes to both x1 and x2?
Yeah, so in this case, it would just be that. This is just going to be the targets of the intervention that I'm drawing the edges to.
I'm just asking, how would you draw it? Do you draw a zeta such that it goes to every variable that
Every variable in the intervention set.
yeah, the single zeta goes to every variable being intervened on?
Right, so this goes back to like this, we have this multi-set of, or this collection of sets, right? So the number of interventions that I have, I'll draw that many zeta variables.
So we're defining these FKs, these different looking at the interventions in division, right?
Yes. Yeah. So what we know is, from the definition of this markov equivalence class is that certain things that we don't intervene on, these conditional probability distributions stay the same, but like if I intervene, say on x2 in zeta 1, and I intervene on x2 and zeta 2, I'm not necessarily replacing these with the same value. I could, in this one, replace it with 1, and this one replace it with 0. Any other questions? So yeah, we'll just take this as an example. This is basically what I wrote here, except now I'm going to be intervening on this x3 variable just to show that in general you can have a still unidentified edges. This is a really good choice in this one if we just intervene on x2, because then we could have told the whole graph. Let's say that we don't know what we're doing, we intervene on x3 instead. In this case, the two graphs on the left have the same V structures and the same adjacencies, whereas the graph on the right doesn't have any V structures. So we'll be able to tell these two graphs apart, but not that graph. Another way I like to think about this is, in this case, zeta 1 affects x3, but that effect doesn't propagate. So the marginal distributions of x1 and x2 in both of these left graphs will remain the same. But in general, the marginal distribution of x1 and x2 can change in the rightmost graph. So this leads us to the definition of I-essential graphs. I think we called these essential graphs CP DAGs, or complete partially directed acyclic graphs, in the other lecture, but that's a mouthful.
Yeah, I have more like a conceptual question.
Yeah.
I think when you intervene on a node,
Yeah.
what people were doing is to delete some edges,
Yeah.
right? But now you're introducing more nodes, and if the intervention is not binary, if it's going to take like 100 different values, then this means that you need to introduce 100 different nodes. But
Yeah, so this is a kind of, I think of these interventions on like an as needed basis. It's like, if I'm actually doing the intervention, then I'll introduce a node, but I don't need to introduce these intervention nodes for everything that could possibly be out there. Like if these are continuous variables that require, like kind of continuous.
and what is the benefit of introducing new nodes?
Essentially just to make the make the reasoning a little bit more similar to the reasoning that we used about deseparation in the observational setting. So if you look at the original papers that define interventional Markov equivalence and stuff, there's a few hoops that you have to jump through if you're not going to introduce new nodes. So it's basically a proof technique.
I see. So you cannot achieve the same thing by deleting edges or removing edges.
Yeah, so you can do it with removing edges, but that doesn't happen generally in soft interventions because So this is also a tool that's especially useful in the setting of soft interventions. And I'll briefly note, I realized I actually never put it on the slides. So if I go all the way back to the kind of different types of interventions that we had, I could have added kind of another access, or another thing that you might worry about with your interventions is that you might have what have been called like these fat hand interventions, where you're trying to do something, you're trying to only affect one node, but you accidentally affect a whole bunch of nodes. And this is actually a problem in gene regulatory networks when we try to do knockout interventions. So just because of the way the technology works, there can be also interventions at other genes that you don't know about a priori, like that's kind of not characterized. So beside your target gene. So you can think of also dealing with these fat hand or like unknown target interventions. And adding in these intervention variables also helps you deal with that because you just treat the problem of learning the intervention targets as kind of the same as learning the edges in a graph. Right, so I was discussing this definition for I-essential graphs, which is similar to the notion that we had in the observational setting, where we just look at the whole Markov equivalence class, or in this case, the interventional Markov equivalence class, and we kind of represent the information that we have about it graphically. So what information do we have? We know all of the things in the Markov equivalence class have the same adjacencies. So our bi-essential graph, which is going to be a mixed graph with both directed and undirected edges, will have the same adjacencies as well. And then if we know every graph in the Markov equivalence class has a certain orientation of an edge, then we'll add that to the essential graph. Otherwise, we'll use these undirected edges to indicate our uncertainty about the direction of that edge. And these I-essential graphs are going to kind of, if you think of the sequential setting that we'll talk about later on, they'll kind of be our representative of our current uncertainty about what the true graph is.
The question is the same as the I-equivalence class? Like if you have the representation that's to be more ambiguous on edges, you don't know.
It's more ambiguous than what?
Than directed arrows. But you know where the arrowheads go. Is this the same as the I-equivalence graph you're introducing?
Not exactly. So I think you're talking about kind of the one that's augmented with those intervention variables. Is that right?
Right, because I'm not mechanism. If you were to do more intervention, the space of potential candidates for equivalence just reduces. Right?
Yeah. So the definition of those augmented graphs included that was based on what the true DAG was. So that was augmenting the true DAG with these variables. This is aiming at a different thing, which is taking the whole collection of DAGs and representing them in one way. So you could imagine leaving in, so this was the case that we had on the previous slide where these two graphs were I-Markov equivalent when we had the intervention on X3. And just based on the definition of this edge is the same between them, this edge is different, so that's why that edge is undirected. You could imagine just leaving in the intervention variable. So this is the same as like, this would be the I-essential graph, or this would be the essential graph if you left in this intervention variable. But because we're just kind of storing the state of our knowledge about the relations between the kind of system variables and not the intervention variables, we just don't draw those in. They don't really matter. Great question. So I'm going a little fast. So I think this section is actually pretty long. So it's good that we're starting it a little bit early. So I'll start off probably before the 10:30 mark by going through the first two non-adaptive experimental design strategies. And just to set up what case we're going to be in. So as I said, we're going to be non-adaptive. And then both of the algorithms that I'll be looking at will assume that we have no observational data. So this is kind of the most pessimistic setting where, you know, your bio grad student and your PhD advisor tells you, you have to go out and you have to do kind of full identification of the system, but I'm not going to give you any data that's ever been measured about the system. You just have to kind of completely design a set of experiments that's going to completely identify any possible system that's out there. And if you don't, you know, you don't graduate or something. Yeah.
Is it the difference between no observational data and observational data of one trivial intervention, like just running?
In the adaptive setting, yes, because then you could run the trivial intervention where you just gather observational data. But I think you'll see with these examples that in the non-adaptive setting, because it could be from any of the Markov equivalence classes, you can't just do that one and then decide all of your interventions after. This is like a form of background knowledge. That's a really good. So yeah, this last distinction doesn't matter as much in the adaptive setting because you could just spend one intervention to get observational data. That intervention being don't intervene on anything.
So I might have missed this, but is this background knowledge taking the form of like a partially integrated graph, or like what is the?
Yeah, this will take the form of the unessential graph. So we have infinite observational data, and we can pinpoint exactly the Markov equivalence class, which is represented by the essential graph. And I'm not doing more general background knowledge because that gets better. I'm only going to consider background knowledge in the form of basically infinite observational data. Great. So I covered those three. I'm going to start off. So this is going to be the difference between the two algorithms. The first one I'll present will be in the bounded setting. In particular, we'll actually only allow single node interventions. And then we'll go to the unbounded setting for the second algorithm. Erase what's on the board because I'll probably want to use it. So just as a couple preliminaries, kind of thinking back to the fact that we don't have observational data, we kind of already observed this fact that if only one node in an adjacent intervention, then we'll be able to obtain, if we know that two variables are adjacent, we'll be able to obtain the direction of the adjacency. And then this is usually not a problem in the setting where we have observational data because we already know all the adjacencies. But suppose that we don't know all the adjacencies already, then we need, in order to establish the adjacency, an intervention where both variables are not intervened. Or I'll show you can also determine it by having one of these orientations tests in one direction and one in the other direction. So from Frederick's PhD thesis, we have a result that we'll use for both of these strategies that in order to determine both the presence and absence of an edge, as well as the orientation, these two conditions are sufficient. And in the worst case, you need to fulfill at least one of them. So the first condition, so this is kind of the condition that we'll mostly be working with when we have observational data, is that we need a structural adjacency test, which is fulfilled if we have observational data. And then we just need a structural orientation test on one of the variables. Another way that we can obtain this information is by having an orientation test on each of the variables. So if you imagine, okay, I have xi goes to xj. I want to determine between these three possible graphs. So can I do this if I have an intervention on each of these variables? So if I introduce this zeta1, zeta2, if I introduce it to all of these, right, then we'll be able to tell between all of them because of this V structure. So we'll have a V structure at xj in this case and at xi in this case, and we'll have nothing down here. Can we do it without either one of these interventions. So can we neglect one of them? Say I just want to do zeta 2. Well, if I'm using a do intervention on this graph, then after intervention, this looks exactly the same as this bottom graph because I've cut off the edge between them. So this kind of clarify why one of these is having one of these is sufficient, but it's also in the worst case going to be necessary.
Sorry, can you just clarify the difference between an XI orientation test and a structural orientation test?
Yeah, this is, they should have edited this before. It's just orientation test in either XI or XJ. Yeah. So starting off with single node interventions, we'll ask ourselves, okay, how many single node interventions are sufficient and in the worst case going to be necessary? So this is the thing I was telling you about, your advisor says you have to discover it. I don't care, use the minimal number of interventions, but if you don't discover it, like, you don't graduate. And if just to kind of clarify between all of the different setups that we're going to consider, I'll write them as these optimization problems. So here, what we're trying to do is minimize the size of the intervention set such that for all G, not in like anything, just all G over P nodes, say, the graph will, or the interventions will identify that graph. And then for all of the interventions in this set, the size of the intervention is less than or equal to 1. And kind of maybe unsurprisingly in this case, it's pretty pessimistic. It's going to require P-1 interventions. So to go through the proof of sufficiency, kind of, I guess not too surprising that this is sufficient. Maybe you'll ask why don't we have to do every single variable. So the kind of scheme that we're using here is to say that we have these like 5 variables. We'll just, kind of obvious, we want to do a separate intervention. We don't want to kind of intervene on the same node twice ever. So we'll pick one variable not to do the intervention on. And then, okay, we want to check whether the conditions from the previous slide here are satisfied. So first, we can observe that all pairs of variables will be subject to one of these adjacency tests. So there will be one intervention where they do not occur. So yeah, just take an intervention. If you want for X1 and X2, take the intervention on X3. That will be provide the structural adjacency test for those. And then all pairs of these variables will have at least one member that's going to serve as kind of the orientation test. So the only thing that you really have to worry about is, in this case, the variable that you don't intervene on. But because you've intervened on every other variable, that's going to be your orientation test. Now, okay, is that really the best we can do? Yes, if we don't know the graph. Kind of the most pessimistic thing is that we intervene on all but 2 of the variables, but the 2 that we don't intervene on happen to be the most upstream. So this is, say these are our interventions. We intervene on X3, X4, and X5. And then I just have in these different colors kind of what edges you learn from each of them just by that kind of orientation test rule. What you don't get is the edge between X1 and X2, because if you think back to the meek rules, kind of none of them allow you to orient something that's upstream of yourself. None of these different interventions will be able to tell you anything about whether X1 is affecting X2 or X2 is affecting X1.
The previous example for 2 nodes, it seems that you need 2 interventions.
Yes, I think this needs a quantifier of p greater than or equal to 3 or something.
Oh, right.
The question here should be p greater than or equal to.
No artificial observational constraint based on the main relation.
Correct. Yeah, there's no, so the question was, we're assuming in this setting that there's no observational data and no other kind of background knowledge about the graph.
There is a that assumption.
Okay. Yeah. Yeah. So we are assuming that the true system is a DAG and causally sufficient. There. All right. So in the last few minutes, I'll go through the keeping all these other things fixed. Now just the unbounded case, and we'll see a logarithmic dependence instead of the linear dependence on the number of nodes. Yeah.
Clarify, what is the difference between intervening every single variable separately versus defining one umbrella intervention that changes the values of multiple variables?
Yeah, that's a good point. So the question was, what's the difference between doing like P interventions where I intervene on every single, in each intervention, I only intervene on one variable, and then doing one intervention where in that intervention, I intervene on P variables. So in the case where you intervene on P variables, imagine the do operation. So that type of intervention where I'm setting it to a specific value, then my actual distribution when I do that intervention on all of the variables at once is just going to be point mass at whatever the thing that I set to. It's deterministically going to be a certain quantity, versus if I do it all separately, there's still variability in all the variables that I don't intervene on. So in particular, this is kind of the tricky thing of why in this structural orientation test, we need that we can only identify between variables where one is in the one is in the intervention and one is not in the intervention. Because if I just intervene on everything and cut all of the dependents, it has kind of no relation to the original system. So I can't tell anything about the edges between these variables. Does that answer your question?
Yeah, but what about if you remove the edge from intervention to X3?
Yep.
And if you define, and in an alternative world, you have X1, X2, and X3, but you're intervening on X1 and X2 separately.
Yeah.
There be any difference?
Yeah, there. So here again, you have one distribution where you intervene on X1 and one where you intervene on X2. In the one where you intervene only on X1, if say X2 is a child of X1, then you'll still have X2 being dependent on X1. And I guess not vice versa, but because they won't both be children of each other. In this distribution, X1 and X2 will be rendered independent. So you won't be able to tell anything about the orientation between those two. So maybe I'll kind of, I think I have time to walk through this result. So this is the same optimization problem you'll see is on the previous slide, just without the constraint on the individual interventions that are in this intervention set. And let's start by proving that you can get by with this many interventions. So I'll give you an actual way of picking the interventions that will completely identify any graph. So it's based on this trick of kind of numbering the nodes and taking binary representations of them. I'm using this to denote the binary representation, and then I'm subscripting to kind of tell you what actual position I'm going to be looking at. So if we take the binary expansion of 6, it's going to be like 1, 1, 0, and then the indexes start from the right. So B6 at 1 is going to be 0, and then B6 at 2 is going to be 1. So the idea is to label the vertices from 0 to p minus 1. So I'll start at 0. And then I'll let my intervention be composed of all of the variables that have a 1 in. So I'll pick floor of log p plus 1 interventions, and I'll let every single intervention be defined by just looking at the different nodes and seeing which ones are 0 or 1. And if it's a 1, I'll include it. So as an example, this is on 8 nodes, and I've started from 0, like I said you should. And then if you kind of think about this, this is basically, kind of doing some mod operations. So here, the first thing is going to be on all the odd numbers. And then the second thing is going to be basically, do 0, 0, and then I'll do 1, 1, 0, 0, 1, 1. So I'll keep alternating and increasing powers of 2, which variables are going to be the targets of the intervention. And this final one is going to be on the last 4 variables. And in this case, because I chose floor of log base 2 of p, in this case, there's 8 variables. So that's 3, the floor just remains the same. I get 4 interventions, so I'll have this extra 1 intervention that doesn't hit anything, because all of the 0 through 7, if you take their binary expansions, aren't going to have anything at the 4th kind of rightmost position. So why does this work? The first thing to notice is that because each of the binary expansions of these different numbers is different, there's going to be at least one intervention at the spot where they're different that includes one but not the other. So we're guaranteed to get at least one of these orientation tests. If p has the power of 2, this is pretty easy. So you'll have that empty intervention out there that gives you your adjacency test for all pairs. And then if p is not a power of 2, it's a little trickier to analyze, but not too bad. So the fact that p is not a power of 2 means that every number is going to have at least 1 0 in this binary expansion. Because if I had all 1s, then I would have to be going up to, in this case, the binary expansion of 7 is all 1s. I would have to be going up to, in my numbering, right below the power of 2. So all of them are going to have a 0 in their binary expansion. And then I have to ask myself, okay, I have my structural orientation test. Now I need either an adjacency test or the opposite kind of structural orientation test. So let's say, for sake of contradiction, that there was a pair of variables where we didn't have either of those. So let's start with saying that we didn't have the adjacency test, so where they're both 0s. In that case, so I write out one of my numbers. I know that say this is the first number, A1 and A2. I know that they have a difference here, and then I, just by the fact that they're different, I'll take A1 to be the one with a 1 in this position without loss of generality. And then I know that I've said there are no, wherever these are 0, this will not correspondingly be 0 if I'm assuming that there's no adjacency tests. So these have to be 1. And then if I assume that they are also no zeros in this one, so none of these opposing ones, then these also have to all be 1. And then I achieve the contradiction of having this number actually has to be all 1s, whereas I said that all of them have at least 1 0 in there. And then, so we're a couple minutes past time, so we'll do the proof of the fact that this is in the worst case necessary after the break.
Okay, great, thank you. We're going to take a break now, as usual, until 11. See you all back then.